Hello, everybody. In this lecture, we're going to talk about DNA replication again. In this case, we're going to specifically talk about elongation and a little bit about termination. This is a semi-complicated process, so definitely make sure you're paying attention here. Rewatch the video as necessary because we're going to talk about some complicated concepts. So let's get right into it. So by the end of this module, you should be able to describe how the directionality of DNA impacts replication and compare and contrast DNA replication on the leading and lagging strands. And so to refresh you on what you have already watched, you have the beginning of replication in prokaryotes and eukaryotes is at the origin of replication. In prokaryotes, there's one origin. In eukaryotes, there are multiple. And DNA gyrase is going to come in and relieve that supercoil stress, which is going to allow other enzymes access to the DNA. DNA helicase is going to unzip the double strands. Your single-stranded binding proteins are going to keep the two strands from going back together. And then your primase is going to add primers because at the beginning, there are no complementary base pairs. And DNA polymerase requires a OH group to bind to. RNA polymerases like primase do not. And so let's get into elongation. Now, our primary DNA polymerase enzyme is DNA polymerase 3. The numbering for these polymerases are based on when they were discovered, the order in which they were discovered, not necessarily the order in which they are used in replication. So this is the main enzyme that's used in DNA replication. It does require that 3' OH group to build DNA. That OH group that it starts with for its very first base to be added comes from the primers, which are added by primase. Now, this enzyme works only in the 5' to 3' direction. And so you're going to take, moving down the line, or if we're going to look at the strand, we go from 5' to 3' in that direction. And on the complementary base, complementary strand, you're going to go 5' to 3' like that. So it always moves in that direction. Now we have two strands of DNA when we begin replication. Those two strands are called the leading strand and the lagging strand. Now, the leading strand is pretty easy to understand overall because DNA is replicated in the same direction that DNA is being unwound. And so it proceeds towards this opening fork as we replicate. Now, one thing that that you see here before we go any further is we see these 3 phosphates that are on our NTPs. This is what we mean when we say NTP. It's a nucleotide triphosphate. Keep in mind when it gets added, 2 of these phosphates get cleaved and you end up with just one. So you have just a single phosphate. And that cleavage of those 2 phosphates is what provides you with the energy to make the bond between this OH and this phosphate over here. And so you start with a double-stranded DNA. It's anti-parallel, so this end goes 5' to 3'. This end goes in the anti-parallel direction, so it's going to be from left to right, 3' to 5'. But keep in mind that everything has to be replicated in a 5' to 3' direction. So our helicase enzyme is going to come through and unzip the 2 strands to form this fork, right? It's going to do this in opposite directions, so you actually end up with a replication bubble, but we'll think about it from the side of one fork. Now, this top strand up here, we've got 5' to 3', and on this bottom strand, we have 3' to 5'. And so, And so DNA replication proceeds in that 5 to 3 direction. So in this top strand, when you add new bases, they're going to be anti-parallel. So the 5' end is going to be of the complementary strand is going to be on this side, and it's going to go that way. So it'll be 5 to 3. So essentially, the complementary strand is going to look just like the strand on the bottom. But the strand on the bottom is going to have a complementary strand that looks just like the one on the top. And so this one down here is going to be our leading strand. And the reason that it's a leading strand is that DNA replication can proceed in the direction of this arrow. And the direction of this arrow is the same direction that helicase is unwinding the replication fork, or unzipping that replication fork. And so this one is going to go this way, while up here, the lagging strand is going to go the other direction, because the new bases are going to always be added 5' to 3'. Now remember, the first bases have to be added using primase. They add a primer, right? And so that primer is going to leave a 3' OH available for DNA polymerase to add bases to. The primase does not require a 3' OH to begin, but DNA polymerase does. Now, when the primase adds its bases, it's going to use RNTPs, like ribonucleotide triphosphates, and they're going to always be added in this 5' to 3' direction. So this purple color represents the new bases that are being added, and so primase is moving in the direction of this arrow. Now, when it ends, it's going to leave that free OH group at the end of your primer. And then primase is going to pop off after it's laid down its primer. Primers are somewhere in the range of like 10 to 20 base pairs. And that's going to give a place for the DNA polymerase 3 to bind onto. So if there was no OH group there, it wouldn't be able to add bases. And so that's why that primer is required. And so DNA polymerase 3 is our primary DNA polymerase. And it's going to start polymerizing in that same 5' to 3' direction. It's going to take up these DNTPs now and add them to the new strand, moving in the direction that the fork is opening. However, we've still got these primers. Oops. We've still got these primers on here, and those primers are made of RNA. We can't have RNA in our DNA strand. And so we have to have a way to get that out. And the way that we do that is with DNA polymerase 1. DNA polymerase 1 has exonuclease activity. And so what that means is it's able to cut out those RNTPs, those RNA bases. And that's what we see leaving here. And then it's going to add in DNA bases in that same 5' to 3' direction. So it's going to cut out one, replace it with a DNTP, move over, do the next one. Okay. Now notice that there's a break that's left between these two strands of DNA, or these two like sections of the DNA. So they were unable to make this bond together. And so this is what's called a single-stranded nick in DNA. And so we use ligase, which is going to bind to either side of that single-stranded break. And then it's essentially going to glue them together. And what that really means is it's going to provide the energy necessary to make a bond between the 3' OH and the 5' phosphate. So then we've repaired that, and you're done. Like, your leading strand just keeps doing this. You know, the polymerase 3 just keeps on going till it meets up with the end of the DNA. Now, the lagging strand is a little bit different. So this strand is made in the opposite direction that the helix is unwinding. And so it proceeds away from the replication fork. So as we see, the replication fork is opening this way, but on the lagging strand, DNA replication is going to go 5' to 3' this way, right? So this way is the opposite direction that it's opening. And so it starts out the same way, right? Helicase is going to unzip that double-stranded DNA. And replication always proceeds in this 5' to 3' direction. So on this bottom strand, this is the leading strand. It proceeds in the direction that the fork is opening. However, on the top strand, which is our lagging strand, it proceeds in the opposite direction from the direction that the fork is opening. So this allows our leading strand to replicate continuously. DNA polymerase doesn't have to stop replicating. It's just waiting for helicase to unzip some more. And as it unzips, polymerase adds new bases. However, for our lagging strand, it's going to have to wait until enough of this fork has opened up for primase to attach on, because primase is going to go in that opposite direction. So it's going to go, it's still going to be 5 to 3, but it's going to go this way, right? And so it's got to open enough for primase to get on. And when it does, it's going to add its primer just like normal. It's going to leave that OH group where DNA polymerase 3 can come in and start adding its NTPs. But we see that now DNA polymerase 3 is back to the origin of replication, and it's got to replicate all the stuff back this way. And so once it hits the other fork, this DNA polymerase 3 is done replicating. So it's going to pop off. And we need to actually open the fork up more before we can do any more replication. And so what that means is as this fork opens up, we're going to get another primase to pop on and add our primer. Polymerase 3 will add to our 3' OH and start adding DNTPs. And then that'll make a new fragment. Then primase will attach down here. It's going to add a new primer, so forth and so on. Now, these fragments are called Akasaki fragments. That's named after the guy who found them. This is one of the rare occasions where your biochemical stuff is named after a person. So Akasaki is the person who figured out how replication on the lagging strand proceeds. And so as you can see, this is a discontinuous type of replication. The only way to do a new fragment is to open up the fork more. If the fork doesn't open up more, then you can't do an additional fragment. Now, eventually, your DNA polymerase 1 is going to come in and replace all of these primers with DNA, which is going to leave you with a bunch of little single-stranded nicks or breaks, right? And so that means that ligase is going to come in and bind to either side and then ultimately repair those nicks by forming the bonds between the 3' OH and the 5' phosphate. Then you get a whole DNA strand. Now, we call this process of DNA replication to be semi-conservative, and that's because at the end, when we have our replication finished, we're going to have 2 DNA helices. One is going to be original to your parent strand, and the other is going to be newly formed. So that's what we kind of see here with our blue and our green. So the blue is our original strand, and the green is our newly replicated strand. It's semi-conservative because we keep half of the DNA left over when we make our new strand. All right. And I mentioned this before, but DNA brings its own energy. Those DNTPs are triphosphates. When polymerase adds them, they cleave off those 2 terminal phosphates, which are known as pyrophosphate, to generate the energy needed to make that 3' to 5' bond. And so here we're going to switch gears a little bit. When our 2 forks come together at the end, you're actually going to have this terminus of replication where you have 2 strands of DNA that are concatenated, which means that they're linked together. And we need those 2 strands to become basically be disconcatenated. And so to do that, we're going to have to break those bonds. And so this is something that is a problem that really we only have to deal with prokaryotes, because with eukaryotes, you're going to pop off and you're going to end up with just linear chromosomes. And so what we need is a topoisomerase. Topoisomerases are able to cause these double-stranded nicks. And so this is similar to what gyrase does. And what this one does is it's going to actually nick the two strands, and then it's going to put them back together as two separate circular chromosomes. So in summary, we've got DNA replication, which begins at the origin of replication, forms a bubble, which is two separate replication forks. You have a leading strand and a lagging strand. Replication in the leading strand occurs continuously because the replication fork opens in the same direction that DNA polymerase moves, which is in a 5' to 3' direction. And we have a lagging strand, which is going to occur discontinuously, forming these Akasaki fragments. Those Akasaki fragments are going to require several small sections of DNA to be replicated. And then all the primers have to be replaced, leaving you with a bunch of individual nicks in the DNA, which are repaired by your DNA ligase. And ultimately, topoisomerase 4 is going to come in and separate your two concatenated DNA molecules. So prokaryotes and eukaryotes are a little bit different in this. We're going to focus primarily on prokaryotes, which is what we just talked about, although we will talk a little bit about some of the stuff that happens with eukaryotes here. Specifically, replication takes longer. It also has multiple initiation sites, while bacteria and archaea have a single origin of replication. And that's all we're going to discuss about the differences here. We do have some other differences we're going to talk about between eukaryotes and prokaryotes in a future lecture. So with that, you should be able to describe how directionality of DNA impacts replication and compare and contrast DNA replication on the leading and lagging strands. All right, I hope you enjoyed, and I'll talk to you soon.